Hello there. Welcome to the Technovation podcast. Thanks for joining us.
Okay, let's think about the deadliest animal on Earth. What comes to mind? A shark. Maybe a lion.
Probably something big with teeth, right?
Exactly. But what if I told you humanity's most dangerous predator, responsible for maybe billions of deaths throughout history, is the mosquito?
That tiny little insect. It seems almost unbelievable, but it holds the grim title.
Yeah. The deadliest animal on the planet.
And today, we're taking a deep dive into how this, tiny terror acts as the primary host for malaria.
A disease that's arguably the single biggest killer of humans in recorded history. It's, truly devastating.
But here's the twist, the really interesting part. We're also looking at a revolutionary new technology, one that could potentially help us eradicate malaria forever.
And this isn't science fiction anymore.
No, the modified mosquitoes, they already exist in labs, waiting for, a decision that could change everything.
So our mission here is to unpack the brutal reality of malaria, first off. Then explore the incredible science behind this genetic engineering method called gene drive. And inevitably confront the really profound ethical questions this power raises.
Absolutely. We'll get into whether malaria is, bad enough to risk it, and what this might mean for fighting other diseases too.
Okay, let's start by really trying to grasp the scale of the problem. We're not just talking about itchy bites here, are we?
Not even close. Think about 2015 alone. Hundreds of millions infected. Almost 500,000 people killed.
500,000, mostly children, right?
Predominantly children under 5,
Yeah.
It's heartbreaking. Imagine a city roughly the size of, say, Kansas City just gone in 1 year.
And it's not some distant problem happening elsewhere. It's a global catastrophe.
A global catastrophe causing unimaginable suffering, especially in vulnerable communities. The WHO estimates, it's claimed more lives than all wars and plagues combined.
That's almost impossible to comprehend. A relentless killer for millennia.
And what's fascinating in a, horrifying way is the sheer brutality of the parasite itself, Plasmodium.
The actual cause of malaria.
Right. These are single-celled microorganisms, and they're completely dependent on mosquitoes to complete their life cycle and get to humans. It's a devastatingly effective partnership.
How does it work? When the mosquito bites?
Okay, so an infected mosquito bites you. Thousands of these tiny early-stage parasites, they're called sporozoites, get injected right into your bloodstream from the mosquito's spit, basically.
Okay.
And they don't just float around, they immediately head straight for your liver cells, like tiny guided missiles.
Into the liver. And then what?
Once they're inside the liver cells, they kind of go into stealth mode. They multiply silently, rapidly, basically hiding from your immune system.
For how long?
Anywhere from a week to maybe even a month. And during that time, they're maturing, transforming, actually consuming the liver cells as they grow.
Oh, wow.
Yeah. Then they change into their next form, thousands of these little droplet-like invaders called merozoites. And then they burst out.
Burst out of the liver cells.
Burst out, flooding your bloodstream. And from there, they launch this relentless attack on your red blood cells.
The cells that carry oxygen.
Exactly. And what's particularly insidious is how they avoid being caught. They infect a red blood cell, multiply inside it, and when they burst out again, they often wrap themselves in bits of the cell membrane.
Like camouflage.
Precisely. Using the victim's skin as camouflage to evade your body's defenses before they go on to invade more red blood cells.
Yeah.
It's brutal. Killing from the inside, then using the remnants to hide.
That is terrifyingly efficient. And this cycle, this invasion of red blood cells, that's what causes the actual symptoms, right? The awful flu-like stuff.
That's exactly it. The high fevers, the horrible chills, the sweats, pounding headaches, convulsions sometimes, vomiting, diarrhea. It's your body reacting violently to all these parasites bursting out and the toxic waste they release.
It sounds absolutely horrific, not just flu-like.
It's much worse.
Yeah.
And it's a cycle. Repeating over and over, escalating the damage each time. Every burst releases more parasites, more toxins, triggering that huge immune response.
And in severe cases.
In severe cases, especially if the infection manages to get past the blood-brain barrier, it can cause cerebral malaria.
Which leads to?
Coma, severe neurological damage, and often death. It's just devastating. And the whole process is perfectly designed so that when another mosquito bites an infected person, it sucks up these parasites.
And the cycle starts all over again.
Exactly. Ready to infect the next person.
And we mentioned it's not just malaria. I remember the Zika crisis in 2015 spreading so fast, causing terrible birth defects.
Also carried by mosquitoes. Different species, but still mosquitoes. Same goes for dengue fever, chikungunya, West Nile virus.
So these tiny insects are just incredibly efficient disease carriers, like perfect parasite taxis.
You could say that. They are truly formidable. They've been around for what, at least 200 million years? Way before us.
Wow, and there are trillions of them.
Estimates are around 110 trillion globally. A single female can lay maybe 300 eggs at a time. Their sheer numbers, their adaptability, their long history, it makes them practically impossible to just wipe out using traditional methods.
Like pesticides or draining swamps, that kind of thing.
Yeah. We've been trying for decades, centuries even. It's a constant uphill battle, often with pretty limited temporary success.
Okay, so if we can't just get rid of the mosquitoes, what if we could change them? Disarm them from the inside.
Now you're getting to the really interesting part. This is where modern biotechnology, specifically a tool called CRISPR, comes in.
CRISPR. I've heard of it. Gene editing, right?
Exactly. For the first time, really, we have the tools to make fast, large-scale, and incredibly precise changes to the genetic code of entire species.
So instead of just swatting or spraying,
what if we could fundamentally alter the mosquitoes that transmit malaria, for instance, making them incapable of spreading it?
how does that work? With CRISPR?
So CRISPR, it stands for clustered, regularly interspaced, short palindromic repeats, but basically think of it like molecular scissors.
Okay.
It lets scientists precisely cut DNA, edit it, even insert new genetic material, like a highly accurate find and replace function for the genetic code.
And scientists have used this on mosquitoes.
They have, successfully. They've created a strain of mosquitoes that are actually immune to the malaria parasite.
How?
They added a new gene, an antibody gene, into the mosquito's DNA that specifically targets the Plasmodium parasite. So if these engineered mosquitoes bite someone with malaria, the parasite can't develop inside them.
Meaning they can't pass it on.
Exactly. They break that brutal partnership we talked about.
But okay, you make some in a lab. How does that help against trillions of mosquitoes out there? Wouldn't the new gene just get diluted out?
That's the crucial problem with standard genetics. You're right. Normally, genes come in pairs, one copy from each parent. So if you breed an engineered mosquito with a wild one, only about half the offspring get the new gene.
Right, standard inheritance.
And with those odds, after just a few generations, the engineered gene would basically vanish in the wild population.
Yeah.
It's like trying to dye the ocean with a single drop of blue ink. It just wouldn't work on that scale.
Okay, so that sounds like a dead end.
It would be, except for this extra twist called gene drive.
Gene drive? What's that?
Gene drive is the mechanism that solves that inheritance problem.
Well,
It's a way to force the engineered gene to become dominant. It essentially ensures its own inheritance, overpowering the original version of the gene almost completely.
how does it do that?
It's clever. The gene drive system includes the gene you want to spread, plus the CRISPR machinery itself. So in an offspring that inherits one copy of the gene drive and one normal copy, the CRISPR machinery in the gene drive cuts the normal copy. And the cell's repair mechanism uses the gene drive copy as a template to fix the break, effectively converting the normal gene into another copy of the gene drive.
Whoa. So it edits the other chromosome.
In the reproductive cells, yes, meaning that parent, even though it only got one copy initially, will pass the gene drive to nearly all of its offspring, not just half.
Okay, that's a game changer. What's the success rate?
Research indicates it could be incredibly high, maybe up to 99.5% of all offspring from an engineered mosquito carrying the drive will also carry the anti-malaria edit.
99.5%. That's staggering efficiency.
It is. So the proposed plan is, well, straightforward in concept. Release enough engineered male mosquitoes into the wild.
Males, because they don't bite.
Right. They'd mate with wild females. And because of the gene drive, virtually all their offspring would inherit the malaria-blocking gene.
And then those offspring grow up and mate.
Exactly. And they pass it on to almost their offspring. The gene spreads exponentially through the population, not killing the mosquitoes, but making the malaria-blocking trait a permanent fixture.
Rendering the population harmless, at least regarding malaria?
That's the idea. If you connect this to the bigger picture, Plasmodium effectively loses its transmission vehicle, its home base.
Its taxi service is out of business.
Pretty much. And scientists hope this change would spread so fast, so effectively, that the parasite wouldn't have time to adapt or evolve resistance.
So, in theory, malaria could virtually disappear.
That's the potential, saving millions of lives. And when you think about the human cost again, that 500,000 children dying every year.
It's hard to ignore.
It really is. I mean, since we started this chat, maybe 5 or 6 kids have died from malaria. For many scientists, the argument is strong. The suffering is immense. It's ongoing. Maybe we should use this technology sooner rather than later.
And you mentioned this might not just be for malaria.
That's right. Malaria could just be the beginning. Remember dengue, Zika, carried by different mosquitoes.
Right.
And beyond mosquitoes, you have ticks transmitting Lyme disease, tsetse flies carrying sleeping sickness, fleas carrying plague.
So the gene drive concept could be adapted.
Potentially, yes, with appropriate modifications for each species and disease vector. Imagine saving millions more lives, preventing just untold suffering, lifting the huge economic burden these diseases place on communities. It's a vision of a world really fundamentally changed for the better.
It sounds incredible. Almost too good to be true. Which always makes you ask, what's the catch? What are the risks?
Yeah, it's a powerful vision, but it absolutely comes with profound questions. Wielding this kind of power, changing a species, potentially an ecosystem.
What stands out to you? What are the big ethical hurdles?
Well, first, why haven't we done it yet if the potential is so huge? Part of it is that CRISPR editing itself is still quite new, barely a decade old, really.
Things are moving fast, though.
Incredibly fast.
Yeah.
But the scientific community is rightly exercising extreme caution. And beyond just the newness, there are these really valid, deep concerns. We have never, ever consciously changed the genetic code of a free-living organism on this scale, intending for that change to spread through the whole wild population.
It's unprecedented territory.
Completely. And the biggest concern is probably irreversibility. Once you release a gene drive, there's no undo button. It's designed to keep spreading.
So you have to be absolutely sure.
You do. And that raises the question of unintended consequences. What if we set out to edit nature and something goes wrong?
Like what? What are the specific worries?
Well, for instance, could the gene drive accidentally spread to other non-target mosquito species? We think it's unlikely because mating is species specific, but it needs testing. Or if you make Anopheles mosquitoes unable to carry malaria, does that create an empty niche that maybe another worse insect fills?
An ecological vacuum effect?
Possibly. Or maybe these specific mosquitoes play some subtle, unforeseen role in the food web. Maybe bats or birds rely on them more than we realize.
How likely do scientists think those risks are?
For the malaria mosquito, Anopheles gambiae, many scientists think the ecological risk is relatively low. They aren't a keystone species. They're a small fraction of total mosquito biomass. Other things eat other insects. But these are critical questions that need rigorous study in contained environments first.
So it's not just about whether the gene edit works, but all the potential ripple effects, the unknown unknowns.
Exactly. And scientists are taking this incredibly seriously. Lab studies contain trials, ecological modeling, trying to understand all the potential impacts before any release. For this specific malaria modification, just adding an antibody gene, it doesn't massively change the mosquito itself.
Okay.
So the worst case scenario in this instance is probably that it just doesn't work as well as hoped, or maybe the parasite adapts somehow. But the broader ecological questions for gene drive in general, those are the really long-term unknowns we're grappling with. We're stepping into a new domain.
Which brings us back to the ethics.
And the moral dilemma. It's a huge debate. Technology this powerful needs extreme care, transparency, global agreement on how to govern it. But you have to ask, is it unethical not to use it when over 1,000 children die every single day from malaria?
That's the terrible weight on the other side of the scale. How do you balance immediate certain suffering against potential uncertain future risks?
That's the core question. And it's not just for scientists to answer. It needs ethicists, ecologists, public health experts, policymakers, and crucially, the communities who actually live with malaria day in, day out.
You mentioned the public discussion feels like it's lagging behind the tech.
It often does with rapidly advancing science. Education and really transparent dialogue are essential here. This isn't just a technical decision, it's a societal one with huge global implications.
So humanity needs to have this conversation, collectively.
We really do. We need informed public debate, international cooperation, strong regulations, clear accountability. We need to figure out how to act on this in the next few years because the power is here, or very nearly here. Our ability to edit life demands an equally evolved ability for collective wisdom.
So summing up, we've looked at the devastating impact of malaria, this ingenious, almost sci-fi science behind gene drive, and its massive potential to reshape our fight against disease.
Huge potential. But as we've discussed, with that power comes this immense, unprecedented responsibility. And maybe the deep dive leaves us with this really profound question to think about. As we get this ability to fundamentally rewrite the code of life in the wild, what does it mean for us to choose which bits of nature to edit? and which to leave alone.
How do we weigh the definite suffering of today against the unknown possibilities and risks of tomorrow?
Exactly. It's not just science anymore, is it? It's philosophy, it's ethics, it's about our future relationship with the natural world. And it's a conversation we absolutely need to have, and probably quite quickly.